Hi, and welcome to our video on Cell Cycle Explained. I'm Hannah.
And I'm Ellie. Let's get started. So first we're going to be talking about the cell cycle, and the cell cycle is just basically all the phases of a cell's life and how they cycle through it. So first we see most cells spend all of their time in interphase here, where there is no cell division happening, and like I said, most cells spend most of their lives here, except for cancer cells, but we will talk about that later. And so we start off here, and every cell starts off in the G1 phase, and this is the longest phase of the cell cycle, and it is where organelles such as ribosomes and proteins are created, which are very important to a cell's well-being. And so a regular cell that wants to continue to grow will move this way, clockwise through the cell cycle, but some will leave the cell cycle and go to what we call G0, which is where there's no more cell division, they're done growing, and an example of this would be neurons. And so next we move into the S phase, which is basically DNA replication. This phase is synthesis. S stands for synthesis, and this is where 23 pairs of chromosomes are duplicated to make 46 pairs of chromosomes. Then they will continue to move this way towards G2, which is where the cell is preparing for mitosis. This is where microtubules are produced, and microtubules help to pull chromatids apart in M phase or mitosis. So mitosis is the last little chunk of the pie here, and it's the most popular, I would say, most well known, but it's also the smallest chunk of the cell cycle. So we're going to talk about mitosis in the next couple of slides, but it's basically just active cell division. So here's just a little overview of mitosis. We start here: interphase, prophase, prometaphase, metaphase, anaphase, which you can't see, but it's right down here, anaphase, and then telophase until it moves into cytokinesis afterwards. But you can see the junk here inside the cells is starting to get organized, and then spindles are created, and everything lines up in the middle here in metaphase, and it splits in anaphase and telophase. Everything is just kind of sorting itself out to be finally two separate cells. So first in interphase, we see So our first stage here is interphase, and this is where the cell spends most of its time, like we were saying. But here you can see chromosomes are starting to condense, but they're not quite visible yet. But they continue to become more and more condensed and visible as the cell cycle continues. So here we have prophase. Prophase is basically where specific chromosomes are becoming visible, and they're now aligned as sister chromatids, and they are going to move into prometaphase later, where the nuclear envelope will fragment. Prometaphase is basically the process by which metaphase we begin to see what we call kinetochore microtubules being formed. These are these long blue spindle looking things here, and they are aligned across the centromere, and basically they're starting to move these sister chromatids back and forth because they're eventually going to line up in the middle in our next phase, which is metaphase. So in metaphase is known as the longest stage of mitosis, and it is basically where the centrosomes are now at opposite side of the cells. These things are completely on opposite poles, and now the chromosomes are aligning down the middle, and the microtubules are pulling them across the middle. In anaphase, we begin to see these daughter chromatids being pulled apart down the middle of the cell. So cell division is truly beginning here. Basically, you can see this is called cleavage, where these split down the middle, and these liberated daughter chromosomes are beginning to move towards the poles, towards these ends over here that we were talking about during metaphase. Next is our second to last phase, which is telophase, and basically this is the two daughter nuclei you see here being formed finally, and the chromosomes are becoming less and less condensed until they have eventually will split down the middle and create two separate cells. This is cytokinesis, basically, where there's splitting of the cytoplasm, where in all the other stages we were seeing splitting of the chromosomes and the DNA and the nucleus. Now in cytokinesis, we're seeing the cytoplasm finally begin to split to finish this process out. This is meiosis here. It's basically a similar process except for it doesn't require two parents and it splits by itself.
So now moving on, in the cell cycle, there are several internal and external factors that control cell division. So first we're going to talk about the internal factors. So first we have cyclins and kinases. And cyclins are a group of proteins that are rapidly made and destroyed at certain points in the cell cycle. So as you see here in this diagram, we have a cyclin-dependent kinase plus a cyclin, it equals an activated complex. So these internal factors are what is needed for the cell to properly go through the cell cycle and to make healthy cells. So kinases are enzymes that transfers a phosphate group from one molecule to a specific target molecule. So again, like as you see here, you see the cyclin and the kinases like working together to make an activated complex. Okay, so now we're going to talk about the external factors that control cell division. So 3 of them that I have listed here are anchorage dependence, density dependence inhibition, and growth factors. So here is a little diagram of a tumor. So tumors in cancer is a result of lack of either density dependent inhibition or anchorage dependence, and we'll kind of touch on this a little bit later, but I just kind of want to explain what anchorage dependence and all of these external factors really are. So starting with anchorage dependence, animal cells must be in contact with a solid surface to divide. That is what anchorage dependence is. And then density dependent inhibition is when crowded cells stop dividing. And then growth factors are secreted by other cells and required to start and continue the division of cells. So all of these factors, if any one of them goes wrong or something doesn't work properly, if anchorage dependence doesn't happen or the growth factors can't happen, then it will be a result of a tumor, and which leads to cancer. We just have a couple examples of normal cells versus cancer cells, which is what we talked about in the last slide of how cancer happens is the result of external factors not happening. So here it kind of shows normal cells. So here we have positive growth factors and cells divide, and negative growth factors and there's no division, but still a normal cell. But in cancer cells, there are positive or negative growth factors and the cells divide, but there's no separation between positive and negative growth factors. So then over here we just have a normal cell which has unfixable DNA damage in the center of both the normal cell and the cancer cell. But in the cancer cell, the cell continues dividing. It can't stop because there's no external factors to help that or help the cell to grow and go through the cell cycle normally. Okay, to close out talking about the cell cycle, I just want to talk a little bit about the proteins that happen in what we just talked about, cancer cells and normal cells. So between these two slides, so RB and P53 regulate the cell cycle on a higher level. So P53 is also called the guardian of the genome. So P21 block cell cycle progression and inhibits CDK. And CDK activates DNA replication and mitosis. So if there's P21, the cell is probably not going to be a normal cell, and there could be tumor growth or cancer. RB is a tumor suppressor gene, or it's pretty much made from a tumor suppressor gene. So a defected or loss of function of the RB will lead to cancer. So those are just some of the, I didn't put a slide in on those proteins, but those are just basically some of the proteins that explains how cancer can happen and how tumors can grow in the cell cycle when these proteins and external and internal factors don't happen. I just want to say this concludes the cell cycle explained, and thanks for watching.
I hope that was helpful.
Bye.